Okay, hi everyone. So our next topic in our microbiology course is microbial nutrition and growth. So we're up to chapter 6 now in the textbook. Now, it's important to remember the last few classes we have been kind of surveying different groups of microorganisms. The next few classes starting today are going to look more at microbial physiology. So what are microbes doing and how do they do it? So we're going to start here with microbial nutrition and growth. So first of all, when we talk about microbial growth, growth refers to an increase in numbers of cells, not the size of the cell. So it's not just one cell getting bigger, it's a population getting bigger. So we start with one cell, one divides into two, two into four, four into eight, and so on. So our population gets bigger. Now, as we mentioned earlier in the course, all living things have requirements that they need for growth, chemical, elemental requirements they need for growth. Carbon, hydrogen, oxygen, and nitrogen make up 95% of the dry weight of cells. You might remember that carbon, hydrogen, oxygen, and nitrogen are also part of those top 6 elements required for life. We might remember the sponge elements, phosphorus and sulfur also added there. Nutrition refers to taking in food and converting it into energy that's required for life and to sustain life. Nutrients are the biomolecules that do that. So biomolecules necessary for carrying out cellular functions. That's what's taken in. We can break down nutrients into several categories. First, we have essential nutrients. Essential nutrients is any substance that must be provided to an organism. An organism can't supply it itself. And that's divided into macronutrients, which are those required in relatively large quantities, large amounts, versus micronutrients, which are also called trace elements, which are required in much smaller quantities. Equally important, but just you need less of them. Now, one of the interesting things that we can do with microorganisms is look at how they perform metabolism and what they're doing with nutrients. And one way to visualize that is with a device called a Winogradsky column. This is a device where we can culture a wide diversity of microorganisms. Basically, it's a column, you put this in a tube, a column of mud from, say, the bottom of a pond or a wetland somewhere. Mix that with some water, some calcium carbonate, and a little bit of sulfur. And then you let that sit right in the window. Let that sit in the sun for several months. And after that point, you'll get a whole diversity, a wide variety of different aerobic and anaerobic bacteria. That's those that require oxygen and those that don't. You'll get some cyanobacteria. That's bacteria that can photosynthesize, and even some algae in there. And what's cool about it is the different types of species separate into layers based on how they're taking in nutrients and how they're surviving, what their metabolism is. So it may look like this. So here we see an example of a real one here, and you can clearly see the layering and sort of how those layers separate out here in the diagram. So the anaerobic bacteria that don't require a lot of oxygen are more at the bottom here, and those that do require light and oxygen up at the top. So that's a way of visualizing these different nutritional and growth and metabolic requirements of microorganisms. Now let's take a look at those in a little bit more depth. So we'll start with carbon here. Carbon, as you might recall, forms the structure of organic molecules. Carbon's the structural basis of life. Carbon is a very friendly atom. It readily bonds to other atoms to form complex molecules, large molecules we call macromolecules. Now remember, heterotrophs are organisms that have to eat other things. So these are organisms that consume other organic carbon sources. So these heterotrophic bacteria here are feeding on the nutrients in the agar on this plate. All right? Heterotrophs, from an ecological perspective, are also called consumers. Autotrophs, again you may recall, are organisms that can make their own food. So they use carbon dioxide, light energy from the sun, and some water from the ground to actually make their own food. And we see purple sulfur bacteria are capable of doing that, and the cyanobacteria here. Ecologically, we refer to autotrophs as a producer because they are producing their own food. Heterotrophs are consumers. They're eating other things. The carbon cycle is a way of looking at the flow of carbon through an ecosystem. How does carbon get into a system? How does it move through that system? And how does it exit the system? So the carbon cycle would include plain old carbon, carbon dioxide, and other carbon-containing compounds. Remember, carbon is the structural basis of all life. We can say life is carbon-based, so it's essential. So this little diagram here just shows us a little bit about the carbon cycle and how carbon flows through an ecosystem. Now, part of the carbon cycle involves organic matter that decomposes and decays. So organic matter can accumulate in the earth over millions of years. And as organisms die, plants and animals and other organisms die, that organic matter accumulates and it begins to decompose. Under the right conditions, this can lead to the formation of fossil fuels. This is a process called carbonification. Basically, layers of decomposing plant and animal matter are exposed to intense heat and pressure as they build up. So this organic matter starts to sink, it's decomposing, it's exposed to pressure and heat underground, and that leads to the formation of fossil fuels. So much carbon is tied up in fossil fuels, coal, oil, and gas. Coal is primarily dead plant material that's accumulated and compressed and been heated over millions of years, where oil and gas is more dead animal matter. So organic matter, as part of the carbon cycle, is involved in this carbonification. Now, another area where carbon can accumulate is in permafrost. So we also have accumulation of organic matter in permafrost. Permafrost is ground that remains frozen all year. This is primarily found in northern climates, so toward the North Pole, okay? Now, permafrost, because the ground remains frozen all year, can store a tremendous amount of carbon because all that organic matter has accumulated in the ground, and because the ground's permanently frozen, that organic matter is very, very, very slow to decay. So that organic matter is stored in that permafrost without decaying for a long time. It's estimated that there's currently more carbon frozen in the ground in permafrost than in the entire atmosphere. And again, this was formed from frozen organic matter that was either never decomposed or very, very slowly decomposed. And this really dense organic matter, really rich in carbon, is called peat. It's very compact and contains high organic matter content. And because of both of these, peat can store a lot of energy. This is what it looks like here. heat soils, and that can just burn and burn and burn. It's storing a lot of organic matter. An issue we're having now with climate change is melting of permafrost. Now think what's going to happen if suddenly that ground is no longer permanently frozen, okay? Large amounts now of that compressed organic matter is going to start to thaw. And as it does so, it releases large amounts of methane into the atmosphere. Methane is CH4. Methane is also a greenhouse gas. So it's sort of a positive feedback loop where as this compressed organic matter begins to thaw and decompose, methane's released to the atmosphere, which furthers climate change. Now, where does this methane come from? This methane was produced long ago by anaerobic methanogens. Remember, methanogens are archaea, not bacteria, archaea that release and produce a lot of methane, and they don't require oxygen. So long ago, all this methane was produced. But the permafrost is so dense and compact that it's essentially anaerobic. Methanogens are anaerobic. They don't need the oxygen. This was good conditions for them, anaerobic conditions. They produced a lot of methane, but the methane remained trapped in the permafrost because it was permanently frozen until now. So now all that methane's being released. So microbes play hugely important roles in the carbon cycle. They decompose all this organic matter, which releases carbon to the soil, to the water, and to the atmosphere in the form of CO2. Many kinds of bacteria and fungi are involved here in decomposition. Xenobiotics. Xenobiotics is another role that microbes can play here. Xenobiotics refers to human-produced chemicals that are not subject to microbial decomposition. So good examples include plastic. Many industrial chemicals are pesticide, but think about plastic. This is a human-produced chemical. It does not decompose. So xenobiotics can have serious consequences to humans and the environment because they're not subject to decomposition and they're not subject to, they're not going to break down and release that carbon back to the atmosphere or the ground. Now, for example, if we look here, what we have here is two examples. This is Roundup. So if we look at the black line, now we read this graph by looking here. The x-axis is time, so as days go by. The y-axis is the relative concentration of the chemical in the environment. So if we spray Roundup here, you notice that over time it dissipates. So over time, the Roundup is subject to decomposition. It dissipates in the environment. So that's not a xenobiotic. But look at Agent Orange here. Agent Orange was a chemical used during the Vietnam War to defoliate dense jungle areas. But you'll notice it's sprayed and it stays at the same concentration over time. So this is what we mean by a xenobiotic, not subject to decomposition, so not part of that very, very important carbon cycle. Okay, now what about nitrogen next here? Remember, the air is about 80% nitrogen. Okay, most of that nitrogen in the air is not accessible to living things directly. It has to be converted to another form first. Now, why is nitrogen important? Nitrogen is a key component of proteins. All proteins have nitrogen in them, and DNA and RNA, nucleic acids. All those have nitrogen in them. So you see amino acid structure, there's always nitrogen here, and in DNA and RNA, look at all the N's over here. Okay, So lots of nitrogen in both of those molecules, very, very important. Nitrogen is also essential for growth of almost all organisms, and growth will often stop due to insufficient nitrogen. So stunted growth, especially in something like a plant, is often an indicator of insufficient nitrogen. Now, as I mentioned, most nitrogen in the air, that 80%, is unavailable to life on Earth. We, for example, breathe it in. If we take a breath, you breathe it right back out. So atmospheric nitrogen is unavailable to most life on Earth. It is, however, available to certain bacteria called nitrogen-fixing bacteria. Now, nitrogen-fixing bacteria are found in the soil, and they can fix nitrogen from the atmosphere. Now, what we mean by fix here is they can take nitrogen from the atmosphere and use it directly. So take nitrogen from the atmosphere, take it into themselves, and use it directly. And what they do is they take that nitrogen from the atmosphere and they convert it to first ammonia, then nitrite, and then nitrate. This is a process called nitrification. And different types of nitrifying bacteria perform each step. So you have nitrogen-fixing bacteria take in the N2 from the atmosphere. Other species will do this step, then this step, then this step. Why is nitrate over here, NO3, so important? It can be used by plants. So plants can uptake that NO3, that nitrate, incorporate it into their body, and then when an animal eats the plant, they get nitrogen there, and something eats that animal, and there's another transfer of nitrogen on up the food chain. So it's atmospheric nitrogen that's pulled in by nitrogen-fixing bacteria. And then eventually we convert to this NO3, which can be taken up by plant roots. Now, nitrogen-fixing bacteria often form beneficial symbiotic relationships with plant roots, and they often grow in little balls called nodules on the roots of some plants, especially legumes, beans and things like that. So you see the little balls here. These is all filled with bacteria. And this is our nitrogen-fixing bacteria here under a microscope. So it grows in nodules on the roots. Now the opposite process, converting NO3 back into plain old nitrogen and sending it back to the atmosphere, is called denitrification. This is accomplished by yet more anaerobic decomposers, bacteria that don't require oxygen. So denitrification, mostly bacteria performing this, some fungi, but mostly bacteria, and we're going to convert NO3 back into N2. So interestingly, everyone, all life is ultimately dependent upon nitrogen fixation. The only way we can access Earth's nitrogen is through these bacteria. So nitrogen fixation is part of what we call the nitrogen cycle, which looks at how nitrogen flows through a system, as we see here. Okay? So our nitrogen in the atmosphere, we have to have nitrogen fixation along the roots, in the soil, nitrification here. We're going to convert to nitrate. That can be taken up by plants. Okay? Now, again, too little or too much nitrogen can have very harmful effects. One of them is called acid deposition, where nitrogen, mainly NO2 nitrites, mix in the atmosphere and then return to Earth as acid rain, snow, or fog, causing effects like this. Look at, here's the original statue in 1908. Look at what that acid rain has done to it. NO2 is primarily an industrial pollutant. You can see that it comes out of smokestacks and so on. So too little or too much nitrogen can have harmful effects. Now, what about oxygen, our next key nutrient here? Now, oxygen's interesting because it goes back a long ways in Earth's history, but wasn't always on Earth. Earth itself is about 4.6 billion years old, but prokaryotic cells evolved around 4.2 billion years ago. So this is the prokaryotes. So about 4.2 billion years ago, you get your first prokaryotes. And this just goes over this here. By about 2.5 billion years ago, we know that oxygen began accumulating in the atmosphere. Okay? So for most of this time, the atmosphere was anaerobic. We did not have any oxygen or very little oxygen in the atmosphere. Eventually, cyanobacteria evolved, okay, in this time period here, okay? And cyanobacteria are photosynthesizers. Now, you might recall that when you photosynthesize, we produce oxygen as a waste. This oxygen got into Earth's atmosphere right here about 2.5 billion years ago. Oxygen begins to accumulate in Earth's atmosphere, forever changing it. So, if we now have an oxygen-rich atmosphere, about 20% of the atmosphere is oxygen, life had to develop ways to deal with what's called oxygen toxicity. Yes, believe it or not, oxygen can be very, very harmful, especially if it's breathed in, taken in its pure form. So if the atmosphere is now oxygen-rich, life has to come up with a way of dealing with that toxic effects of oxygen. So even today, toxic oxygen can form as part of metabolism. Particularly, this is called superoxide free radicals, O2 negative, a negative charge on it. These free radicals, these superoxide free radicals, are highly reactive and would destroy cells if they're just left to build up. So life had to come up with a way of dealing with that to avoid that destruction. So enzymes eventually evolved to deal with this problem. One of them is called superoxide dismutase, or SOD. Now, this is an enzyme that evolved as oxygen built up in the atmosphere and began to become more and more toxic. And this enzyme's job is to neutralize these free radicals. And it does so in this reaction here. So we have our free radical, another free radical, and we have hydrogen. Superoxide dismutase, the enzyme, hooks these together, forming H2O2, which is hydrogen peroxide, and regular oxygen. Now, that's step one in neutralizing toxic oxygen. The problem is this hydrogen peroxide is also toxic. So now we have to deal with this toxic substance. All right? Two enzymes deal with this problem, catalase and peroxidase. Here's a depiction of catalase, the enzyme. Now, catalase works like this. It combines two hydrogen peroxides, and in the presence of catalase, it breaks that into two waters and two oxygen. So now we don't have anything toxic here. Peroxidase works this way. We take our hydrogen peroxide plus hydrogen, and we make two waters from it. So we can test for the presence of these free radicals and the presence of these enzymes by doing what's called a catalase test. Here's bacteria here growing on this agar plate, and if I drip hydrogen peroxide onto the bacteria, we see all these bubbles. So what do you think the bubbles are? Okay, I drip hydrogen peroxide on there, they have to use catalase, and they break that into water and oxygen, so you're seeing bubbles of oxygen be released here. All right, so let's look at oxygen requirements in microorganisms. Now, one of the neat things, and one of the things that I personally really like and appreciate about microorganisms, especially bacteria, is how versatile they are. Prokaryotes, particularly bacteria and archaea, have a tremendous diversity in how they can make energy, how they can take nutrients in, and how they can perform metabolism. They're much more variable and they have a lot more abilities to do that and different ways of doing that than any eukaryote does, as we'll see here. So some microorganisms are aerobes. Aerobes require oxygen to make ATP, chemical energy, and survive. They will die in the absence of oxygen. So most life on Earth, including ourselves, are aerobes. We require oxygen to make energy in the form of ATP, and we die in the absence of oxygen. Other microorganisms are facultative anaerobes. They can grow with or without oxygen. Now, if there's oxygen present, they're going to use it. Because if you have oxygen present, you can make more energy. If there's no oxygen present, they will switch to fermentation or something called anaerobic respiration if there's no oxygen. And we'll be talking about these processes in the next chapter. We also have obligate anaerobes. These are strictly anaerobic and will die in the presence of oxygen. So oxygen actually kills them. They have to stay away from oxygen. Number 4, there are what's called aerotolerant anaerobes, which are strictly anaerobic. They can't use oxygen. They can't switch over to oxygen. They're totally anaerobic, but oxygen's not toxic to them. Oxygen does not poison them. And then lastly, we have what are called microaerophiles, which require oxygen to survive, but are poisoned by high amounts. So they need oxygen to survive, but only a little bit. Too much is going to destroy them. So if we look at these tubes with the dots represent microorganisms, which number goes with which type of microorganism? So you can ponder that, and when you think you have it, here's the key. So number 1, aerobes. Aerobes need oxygen to survive, so they have to be toward the top, toward oxygen. Obligate anaerobes are killed by oxygen, so they want to be way away from it. They're at the bottom. Facultative anaerobes, remember, can live with or without oxygen, but if oxygen's available, they can make more energy. So you'll find them throughout the tube, but mostly toward the top, because they can make more energy with oxygen. Microaerophiles is number 4 here. Remember, they're anaerobic, but not poisoned by oxygen. Okay, so they don't need to be right at the top. They're anaerobics, a little bit below the surface. And aerotolerant, or excuse me, these were microaerophiles here, require oxygen to survive, but are poisoned by high concentrations. So microaerophiles, they need oxygen, so they're going to be near the top, but not right at the top, because high concentrations destroy them. And then the easy one's aerotolerant. That's number 5 here. Aerotolerant is strictly anaerobic, but not poisoned. So they're just evenly distributed. Okay, they don't really care about the oxygen. All right? So you could think about some general questions related to oxygen and microbes. Why can a catalase-negative bacteria, of which there are many, still grow in the presence of oxygen? So in other words, this would be a bacteria that doesn't have the enzyme catalase. So how can it grow with oxygen if oxygen's potentially toxic? Remember, there are 2 enzymes. It's using the other enzyme, peroxidase. Will a facultative anaerobe grow faster aerobically or anaerobically? I kind of told you that one. Aerobically, you can make a lot more ATP if there's oxygen available. Why is a puncture wound more susceptible to tetanus than a more superficial wound, just on the surface? Well, tetanus is caused, we might recall, by Clostridium tetani, which is anaerobic. So it can live at the bottom of that puncture wound away from oxygen. Flooding a wound with hydrogen peroxide, which you might have done, you get cut, you put hydrogen peroxide, results in the production of bubbles. Why, and what if it doesn't bubble? Now, we looked at that picture before. The bubbles are oxygen released from catalase. So what if you put peroxide on that cut and it doesn't bubble? That can be a tricky question. I want you to ponder that, okay? Now remember, all life is going to use catalase, okay, or peroxidase. Catalase is essential for life. No living organism that is exposed to oxygen can live without it. So an organism developing a defective gene for catalase, say you can't make catalase, you'd never be born. Oxygen would kill you from its toxicity. So if you pour hydrogen peroxide on that wound and it doesn't bubble, it doesn't release oxygen, something's wrong with your peroxide, not the organism, because you'd be dead without that catalase. All right? So let's look next now. Those were chemical requirements for microbial growth. Let's look at some physical requirements for microbial growth. And I think everyone, we're at about half an hour here of time. So I'm going to split this lecture, this chapter, into two parts. So let's end part one right now with those chemical requirements. And then I'm going to pick up with part two, physical requirements, here in the next video. So that's it for right now. I'll see you momentarily where we'll do part two of microbial nutrition and growth.